Welcome back, friends. In this video tutorial, we'll be talking about Helicobacter pylori, and especially we are talking about the introductory part about the Helicobacter pylori bacteria. Now, I must tell you, Helicobacter pylori is one of the most significant type of microorganisms that we want to talk about in this whole course, because this is related with a very different kind of and very clever mechanism of causing disease in human being using a very specific chemical enzymatic system, and we'll be talking about the system in details in future. Now let's first talk about the basic features and properties of Helicobacter pylori. Now, Helicobacter pylori are gram negative. Definitely they are gram negative organisms, and they are having a typical structure. The characteristics structure move of the gram Helicobacter pylori is a kind of like this. It's not a straight, it's not a circular, it's not a rectangular structure. Instead, it's a kind of curved structure like that. They are gram negative, but they are having this kind of curved structure. Usually under microscopic field, usually we can see more, or so let's say two or more pylori are arranged like that. They are attached with each other like that. So let us draw something like this. So you can find the structure like that. So these are those bacterias. These are those bacterias, right? And usually, so let me talk about a little bit. Now they are curved or spiral. So let me write curved or spiral in nature. This is the first thing. And second thing is that they are having flagella. And the flagella we are talking about, they are having multiple flagella at the polar region. So let me draw this is multiple flagella in the polar region, right? So that means they are multiflagellate. So multiple flagella in pole, okay, in any one of the poles, right? And but the most important and significant part about this Helicobacter pylori or Helicobacter pylori is that they can produce urease, which is an enzyme, right? Because it is having an ase, it's an enzyme. Now this enzyme is having the property to convert urea into ammonia, right? So it's ammonia gas, right? Now this ammonia can react with water to make ammonium hydroxide and all these things, because this ammonia is basic in nature. Now as it is basic, that means high pH, so it will increase the pH of the medium, right? So this is a very important part. Now this enzyme acts on it, convert urea to ammonia. It is basic in nature, right? Now this Helicobacter pylori, so this is a property, and what is the importance of the property, we'll be studying it later. Now another important property is that Helicobacter pylori lives in, they colonize, so let me write, they colonize in gastric mucosa, gastric mucosa or gastric epithelial cells. So they usually colonize in gastric epithelial cells or gastric mucosas, right? Now what is gastric mucosa? So if I draw our structure a little bit, so let's say here it is, so it's a diagrammatic representation. Say this is our stomach, and right after the stomach we are coming in, and we are having a lot of things going on there, and obviously we are having large things coming out like that. So here it is a kind of structure like that. So if I draw, if I'm right that this is a kind of digestive system structure, in this case if this is the stomach, so let's say this is the stomach and this part is the duodenum, right? So this is the duodenum. Now this region is important, and also this part which is called the esophagus, right? Esophagus. So this is esophagus. These are the two regions where this Helicobacter pylori usually colonizes. Okay, and they usually colonize in the inner side of it. Now usually let if I draw it again, so if this is so let me go to a little bit. Okay, so let's say this is the lining of the cells. These are the cells. Let's say these are the cells. the segment wise cells are arranged. They are having nucleus and all this. These are the cells, and outside of the cell there is a mucosal layer. Now this mucus layer prevents duodenum wall against the HCl, right? So they prevents against acid. Now the acid is coming from stomach, right? So they prevent this duodenum wall against the stomach acid, right? So that's a very important point in all these cases. Otherwise, we know that here in the stomach we are having very acid content, because acid cells, secreting cells, release acid. The acid secretion is important because some of the enzymes require this acidic environment as their optimum environment to carry out the reaction, right? Now in this case, as the acid is secreted, the environment is much more acidic. Now as the environment is acidic, there is a difficulty in the bacteria like Helicobacter pylori to live there, right? Because they need to colonize in these places. So for colonizing of this bacteria in this particular place, they require to low down the pH a little bit. And the way to low down the pH, usually we know that suppose we are having an acid somewhere, right? So that means a low pH. Now if we need to balance this acid, we need to low down the acidic content. What we need to add? We must add base or basic content, right? So if we add base with this acid, in those case only, because we know bases are having high pH, in those case only, right proportion of attachment will provide us neutral condition. Sorry, it will provide us neutral condition or neutral pH, right? So that's very important in this case, and this thing can be achieved using this urease enzyme. The urease enzyme are secreted, not it's secreted by Helicobacter pylori. So that is the importance or magic of urease enzyme secretion. That's why they're secreting this. Now this urease will convert urea into ammonia. Ammonia is basic, so it will reduce down the acidic content. Now the environment will be neutralized, and then the bacteria can easily colonize there, right? So that's the basic funda. We'll be talking about it later in future in the pathogenesis video. And another important thing is that they are related with different kind of gastrointestinal diseases, related with the symptoms like diarrhea, abdominal cramp, and all these things, because they are related with gastritis actually. So if I talk about here, so they're related with gastritis, different types of gastritis, and also they are related with we know it is called ulcer, right? Ulcer means it's a damage of your this in epithelial lining, so it's called ulcer, right? Peptic ulcer. So usually this ulcer is also called peptic ulcer. Okay, so usually this kind of infection, if it is becoming chronic, it can cause ulcer. Otherwise gastritis, different forms of gastritis are caused by Helicobacter pylori. And we can culture this bacteria using different selective mediums in our lab. But this bacteria is growing very fast, and they are using their mechanism to go against our normal defense mechanisms, and they also can recruit other immune cells in this place too, right? So this is a kind of condition. Now in most of the people can have this kind of Helicobacter pylori inside their body, but some of them, very few of them will go to cause several kind of diseases like gastritis or ulcer and all these things. Now, mild form of gastritis can be seen in many bacteria, especially the many people. Sorry, especially in country like India, you can find many people suffering from small or mild symptoms of gastritis in most of the cases, and those gastritis most of the times are caused by Helicobacter pylori. But we are not bothering about that because that are not causing us that much of pain. But now, if it is of chronic case, and if it is growing better and bigger throughout the time, then it may lead to ulcer. In those cases, we need to be very careful, and we need to seek the medical attention, right? Otherwise we don't require it. So that's about the basic properties of Helicobacter pylori. In the future video we'll be talking about the infection. So far we have talked about the introductory video about Helicobacter pylori. Now in this video we'll be talking about a little bit about infection. Okay, so we'll be talking about the type of infection caused by Helicobacter pylori. Now the type of infection they usually causes, they are related with we know different symptoms like diarrhea and different GI symptoms like gastrointestinal cramp and all these things. Now usually the diseases they cause is gastritis. So let me write gastritis, as well as they also cause ulcer, which is also called as peptic ulcer, right? So let me write it. It's also called peptic ulcer. Okay. Okay. And as they're attached with related with ulcers and gastritis, they are also related with symptoms like diarrhea, chronic abdominal pain, and different things like that. Okay. And the disease like gastritis, as a result of this kind of infections, the acid efflux takes place. Acid efflux, acid efflux. Now, this kind of acid efflux will damage. This can damage. Sorry. This can damage esophagus, right? So this is a kind of cause of the disease. And in different modes of ulcer, there are different modes of ulcer, right? Now in gastric ulcers, what it actually causes, it results in, so let me write, it results in inflammation. Results in inflammation or the swelling of this place, right? Most of the case inflammation is related with many things. It is related with physical characteristics or appearance, such as swelling, and also it is related with calling upon the immune system, host immune system. So it will call upon host immune system in the place, right? Now why they are doing so, it is also important, we'll be telling it later. Now another thing that I must talk about this part is that uh, now normally they can also cause this epigastric discomfort and duodenal ulcer. Okay. Now as they're colonizing in duodenum and esophagus, so duodenum ulcer and esophageal ulcer, this can be a cause of the disease. And also sometimes it can be normally these things are what you can say, uh, placed in a particular region, so they are non-motile kind of infection, but sometimes can be spreading. So this can sometimes spread from one place to another place. In those cases they are related with a form of cancer. This thing is important because let's say here it is. So let me draw. Let's say here it is our stomach, and this is the duodenum coming out. Now this is the region for the duodenum, for example. So in this region, suppose these are the bacteria, these red color things are the bacteria that are colonizing there. Now after colonizing there for many hours, many days, not hours actually, colonizing there for many days, what they are producing, they are producing biofilms. Biofilms are generated. Now these biofilms can be transferred as a mass of bacteria. So as a mass of bacteria from that place to other place, from that place to other place. As a result of this movement of this bulk bacterial region from one place to another place, it will be called as metastasis, right? So we can call it as a metastasis. This kind of behavior is a typical behavior for the cancerous cells. So this may turn into a kind of cancer, but it is it is occur only when the infection is going on for longer period of time. Otherwise, there is no chance for this to happen. Okay, and okay, so that's about it, and I hope that's helpful. Thank you. Welcome again, friends. We have already talked about the Helicobacter pylori introduction, and also we have talked about the infections they can cause. Now in this video we'll be talking about, let's say, the pathogenesis of that bacteria. Okay, now for the spread of the disease, every bacteria requires some strategies, right? Now in this case of Helicobacter pylori, they develop a very special and clever way to fight against the acidic content of our stomach, right? The only thing that can survive there is acid loving bacteria or acid loving particles, but usually in stomach the acid content is very high. That means the pH is very low, right? Now in those conditions, if it need to colonize somewhere very near to the stomach regions like duodenum or esophagus, it need to survive high acid content, right? And that is the major thing that need to be done. In this case of this Helicobacter pylori infections, they are overcoming that particular thing using the enzyme urease. That is a blessing for all this Helicobacter pylori. Now this urease enzyme, so let me write urease enzyme. Now what this urease enzyme can do is that, let's say we are having urea, and it can convert this urea into ammonia and also carbon dioxide. So let's say here it is the enzyme acting. So let me draw it. The enzyme is acting on here. It is causing this urea to be converted into ammonia and carbon dioxide. Now this ammonia that they produce is basic in nature. So this basic ammonia will reduce eventually the sorry this basic ammonia will increase the pH of the environment and bring it to the neutral condition. Neutral condition. And that is what is needed. Now in neutral condition, many bacteria like Helicobacter pylori can easily live on. Right? So that is a very important concept about the Helicobacter pylori infections or Helicobacter pylori pathogens. So this is the first important thing. The second important thing is that they call upon host immune cells also. Okay, and this host immune cells also produces urease. As they are also producing urease, this can again, so let me draw it again, this can again go and do the same work that is done by the urea secreted by the bacteria. So they are not only providing the enzyme to degrade urea to ammonia, but also they are amplifying it by calling upon host immune system cells into the place, like macrophages, neutrophils, and all these cells, right? So this approach is very clever, very clever approach, right? Now usually what they can do is that, so if I draw this structure, I've already drawn it in previous videos, so let me draw it again. So let's say this is the lining of the cells. So let's say this is the lining. So gastrointestinal lining of the cells. Cells are placed like that, and below that below that there is connective tissue. So let me draw the connective tissues there. This is the layer for connective tissues, and onto the top of that, this is the epithelial layer, and onto the top of everything here, what we'll be having a mucus layer. This is very important. So three layers. This is a mucus layer. This is, so let me talk about, this is connective tissue. So this is connective tissue, and this middle one is the epithelial layer. Okay, so these are the three layers. Now what is going on, that this bacteria will come. So let me draw the bacteria. So the bacteria will come and sit onto this mucus layer, and as they're sitting onto the mucus layer, they start to secrete that urease enzyme outside. So let's say this is the urease enzyme. They start to secrete this urease enzyme outside. Now this urease enzyme will convert urea into ammonia, but what they require is urea, right? Now wherever we are talking about inside stomach or duodenum, in that region, right? Now there is a question that can come in your mind, that from where our stomach gets urea. Now one possible answer for that is that the urea is also provided by the bacterial cell, but this is highly unlikely because bacterial cell cannot produce such a massive amount of urea in a very few amount of time, because it is supplying urease too, right? And if it is supplying urea, then the urea that is present inside the bacterial cell will be destroyed by the urease that is secreted by itself too, right? So for that reason, that can be secreted by the stomach cells. So that's the one answer, that the urea can be secreted by stomach cells, or other cases that the urea is synthesized by other cells from different regions, and that is delivered to the stomach regions for this activity, okay, or for the activity of normal pH control in uh, inside stomach, or inside duodenum, right? So for that reason, because stomach is having a very low pH, that means very acidic, so for the general time control of the pH inside stomach, our own host body system is developing the plan for secreting urea to the place. So that's the one possible answer for the puzzle, right? Now let's say this is the case, that normally our body is producing urea to low down the pH, to regulate the pH in stomach. But now what they're doing, as the bacterial infection is taking place, the urea is always there, the bacteria is secreting urease, the urease is degrading urea into ammonia, which is a form, and obviously it is basic, and also carbon dioxide. This carbon dioxide will be uh, what you can say, it will come out by the respiratory pathway, and ammonia will be there, because ammonia is basic, it will be dissolved into the solution that is present there, and it will form basic condition. Now the basic condition formed will neutralize the pH of the medium, but it cannot neutralize the pH of the medium inside the whole stomach, because the bacteria is very small, and what they can do, this kind of release of urease will able to create a small environment where they can neutralize. So neutralized, let me write, neutralized environment. Now the bacteria can develop a micro-neutralized environment surrounding it. Now this environment is enabling them to survive for a longer period of time there. As they're surviving there, they can divide and multiply and grow. After a certain time, they start to secrete some components that are going to degrade this mucosal layer, right? As they're degrading this mucosal layer, after degrading the mucosal layer, the bacteria can enter inside. Sorry, so let me change the color. They can easily enter inside. So now as they are entering inside, so let me draw this with this, okay. So as they are coming inside, after going and touching into this cell layer, they start to evade inside the cell, and then finally they can come to the connective tissue. So they can spread from the top layer to the bottom layer like that, right? And this kind of transfer is very important, and during this transfer they also call upon immune system cells. Now cells like macrophages, dendritic cells, and other kinds of cells, those cells will come, and as a result of this calling upon of different host immune system cells and other things, they start the place start to swell, and it will change its color into red. So it, and obviously pain start to occur, and all this thing, and all this swelling, redness, pain, and everything, these things are a sign of inflammation, right? So this is eventually cause inflammation. So it's not it should be inflammation, right? So inflammation will take place in very few amount of time. Now the inflammation, if the inflammation start to grow and grow in higher amounts, it will block your food eating activity for some times, and obviously digestion for some time. So it is a kind of dangerous situation, but because every time you eat some food, the different gastric cells secrete some acids outside, that acid is going to destroy eventually your cell lining, and as this infection is going on chronic way, more and more acid, more and more cells start to secrete acid, and they start to destroy it, and it is finally destroying the lining of your gastrointestinal wall, and finally it will lead to ulcer. So that's what it is called. Ulcer means the damage of the gastrointestinal cells, right? So ulcer means, so let me write, damage in GI tract, right? So that's called ulcer, and this is caused due to prolonged exposure. So let me write prolonged exposure, right? So That's how this bacteria can cause disease, and they can also move from one place to another place. I've already talked about it, and they can cause several kind of infection, cancers conditions, right? So that's it, and I hope that's helpful. Thank you. So we have already talked about introductory parts and infections and pathogenesis of Helicobacter pylori. Now in this video we'll be talking about a little bit about treatment. Sorry, about treatment of Helicobacter pylori or Helicobacter pylori. Now, for the treatment purpose, what we require, as we know that Helicobacter pylori infections are common in GI regions or gastrointestinal regions. Now, as they are related with the environmental conditions, like the acidic environment can kill them, and that's why they are providing the urease enzyme, which will convert urea into ammonia, so that the basic environment will neutralize the environment and all these things. Now, to kill them, one thing we can do is that we can induce, or we can reduce. So let me talk about. We can reduce the proton pumps present in the cell of gastrointestinal lining. So if I draw it again, if these are the gastrointestinal lining cells, these cells are having proton pumps. So let me draw. They are having proton pumps. Through those pumps, so it will look something like that. So let's say these are the proton pumps. So these are, say, proton pumps. Now through this proton pump, proton can enter or exit. Proton can enter or exit. Okay, so usually it exits outside, right? So that if this is the epithelial lining of cells, and these are the mucosa, and this part will be uh, the stomach region, right? So proton will be released outside in all this case as a result of release of the proton outside. So let me draw this. It should be in this direction arrow. So proton can be out. Now if you block this proton pump, it will lead to less release of protons outside. So as a result, our gut lumen, or we can say, yes, our gut lumen will be less acidic, and that will end up with less damaging effect on these endothelial cells after the infection of Helicobacter pylori, right? But the other times this proton pump activity is very, very important. Otherwise, some important proteolytic enzymes will not be functional anymore, because they require the acidic environment to function properly. Okay, now that's one thing that we can do. So for the treatment of the of this Helicobacter pylori infections, what we can use, one way of going it is to use proton pump inhibitors. Proton pump inhibitors, we can use it. Proton pump inhibitors can be used. And the second case is, obviously this is a bacterial infection, so we can use antibiotic. This is always there, right? So if we are using antibiotic, among the antibiotics, among penicillin type of antibiotic, what we can use, among penicillin type, we can use amoxicillin. Okay, let's talk about second one. Among macrolides or macrolides, what we can use, clarithromycin. And among other type of antibiotics, we can use this kind of proton pump inhibitors, right? So this is one of very important, because the infection usually caused by Helicobacter pylori of two different type. One is gastroenteritis or gastritis. Let's say gastritis. This is the first kind. And second kind of infections, or second kind of disease symptom that they cause is ulcer, right? Now ulcer means damage in the epithelial cell line. Now this damage is caused by acid or high acid content, and the high acid content is made using, or it is possible using, activity of proton pumps, right? So if we block, so this is the idea, if we block proton pump, it will reduce the acid content, so it will reduce the effect of ulcer. So that's what we are doing using this proton pump activity. So we can use this type of antibiotics, which are the proton pump inhibitors, and on the other hand we can use amoxicillin and what you can say, amoxicillin and clarithromycin. So it is a clarithromycin, not C, this is a Cl. So clarithromycin or amoxicillin for the killing of the for the killing of this Helicobacter pylori bacteria inside. So in most of the cases there are combinatory therapy using proton pump inhibitors along with amoxicillin or clarithromycin to treat this kind of Helicobacter pylori infections. Okay, so that's it, and I hope that's helpful. Thank you.